Welcome to Mastering Machines, where we discover tomorrow's reality.
A couple weeks ago, at the Google I/O keynote, Demis Hassabis, the CEO of DeepMind, talked about a couple of projects. One of them being, Project Astra, which was an agent framework that they were creating, and then also he talked about AlpaFold 3, one of the premier tools that DeepMind is working on, something that could change how research is completed and done and even accelerated within pretty much every scientific field you could think of. Did you know that there were over 200 million known proteins, with new ones being discovered every year? Each one has a unique 3D shape that determines its function and purpose. But here's the challenge for scientists: trying to figure out the exact structure of a protein. And that can take years and hundreds of thousands or even millions of dollars to crack that code. So that which means scientists can only study a tiny fraction of them. So that slows down research to combat diseases and find new medicines. But what if we had a tool that could change that? And that is the proposition of AlpaFold 3, to hopefully change that. AlpaFold wants to find out the mystery behind these proteins. So imagine a delicate string of beads, each one representing a unique chemical building block called an amino acid. These beads are carefully arranged according to the genetic blueprint encoded in an organism's DNA. So as the string folds, the 20 different types of amino acids interact, attracting and repelling each other in a really interesting kind of dance. This intricate process is known as a spontaneous origami, and it gives rise to the complex 3D structure of a protein, complete with curls, loops, and even pleats. But determining the structure of a protein is no easy feat. And scientists have historically relied on laborious methods like nuclear magnetic resonance and X-ray crystallography, which require years of trial and error and millions of dollars in equipment and a healthy dose of patience. So for decades, the holy grail of protein research has been to, kind of crack that code. And can we actually predict the 3D structure simply by knowing its sequence of amino acids? This is what's been referred to as the protein folding problem, a challenge that has captivated a swath of scientists for generations. The journey for AlpaFold started when it was still AlphaGo, and it beat one of the top international Go players, Lee Sedol. Over the next 4 years, the team focused their efforts on cracking one of the biggest challenges in biology, the protein folding problem. They started by teaching AlpaFold using the sequences and structures of around 100,000 known proteins. This extensive dataset helped AlpaFold learn the intricate relationships between amino acid sequences and their corresponding 3D shapes. Every 2 years, the CASP, the Critical Assessment of Protein Structure Prediction, organizes a challenge for research groups to test their protein structure predictions against real experimental data. And here's how the challenge works. CASP provides teams with amino acid sequences for proteins whose exact 3D shapes have been mapped but are not yet publicly released. Teams then submit their best predictions, which are later compared to the actual structures. In 2018, at an event called CASP 13, AlpaFold secured the first place. But the journey didn't end there. In 2020, at CASP 14, AlpaFold 2 was introduced. The results were nothing short of exciting and revolutionary. AlpaFold 2 achieved a level of accuracy so high that the scientific community considered the protein folding problem solved. The impact of this breakthrough has been profound. The AlpaFold 2 methods paper has already received over 20,000 citations in scientific literature, placing it among the top 500 most cited papers of all time. And that's across any field. Now that we've talked about the history of AlpaFold and how it kind of came to be here, I also want to talk about one of the newer products, or one of the newer tools for AlpaFold, which is called the AlpaFold server. It has been said that this is the most accurate tool in the world for predicting how proteins interact with other molecules within the cell. This platform is available for free and is specifically designed for scientists, and it can be used for around the globe for non-commercial research. And with just a few clicks, biologists can harness the power of AlpaFold 3 to model complex structures composed of proteins, DNA, RNA, and a variety of ions and chemical modifications. The AlpaFold server represents a significant advancement in the field of molecular biology, providing researchers with an unprecedented level of accuracy in their predictions. Just to give you a sense of what are some of the things that AlpaFold has been able to really improve and help as far as on the scientific and medical front is, it's been able to develop a vaccine for malaria, being able to detect signs in osteoporosis in humans, understanding the faulty proteins linked to cancer and autism, and even solving the mystery of an ancient bird that went extinct over 50,000 years ago by looking at the different proteins and all of that stuff with the archaeological skeleton. It's only a matter of time before digital biology, especially with tools like AlpaFold 3, become more mainstream and become entire industries and sectors where we're building molecular elements specifically through digital tools. And obviously that's going to be a pretty crazy time once that happens. But AlpaFold 3 looks like it will be ahead of the pack when it comes to that. Remember, mastering machines, discover tomorrow's reality.